Alright, so again, briefly, we're going to talk about some mutations that can occur in DNA that can affect the overall protein sequence and function of an organism. So we're going to focus here on something called point mutations, and we'll first look at the wild type or the normal type seen in most people. And we have a CTT in the DNA, for instance, for our nitrogenous bases, and then we look at the mRNA that transcribes into GAA, and then that results in the coding for this particular amino acid, glutamine. Now, over here, if you have a point mutation in the gene for hemoglobin, and this occurs in people that have sickle cell syndrome, they have one nucleotide that's different. Instead of a T in position, the second position, they have an A, an adenine. That results in a different codon in the mRNA, a GUA, that results in a different amino acid, valine, and it results in the hemoglobin being structured such that the red blood cell takes on a sickle shape instead of a nice round donut, carry all kinds of oxygen shape. And in these individuals, they cannot carry the same amount of oxygen on their red blood cells or in their red blood cells as someone that doesn't have this point mutation. And it causes a whole series or list of conditions and problems that are associated with this just one base change. Unbelievable. But it happens. So there are different kinds of mutations as far as these point mutations can occur depending on what happens with the amino acid sequence. Here we have our wild type mRNA, the normal mRNA, I guess you could say. And we have, as a result of the mRNA sequence, these 4 amino acids. Now, if we have an A instead of a G here at this position, let's see, 1, 2, 3, 4, 5, 6, 7, 8, 9, at position 10, that results in AGC instead of GGC, results in serine instead of glycine. And when you look at this, you think, oh, there's 4 amino acids, we're good. But you take a closer look and you say, something is amiss here. The wrong amino acid is inserted or is in place here. So that is a missense mutation. When you have the right number of amino acids, but you have the wrong amino acid or amino acids, that is a missense mutation. Now, another type of mutation that can occur from a point mutation would be something called a nonsense mutation. So here we have that same wild type normal sequence with the 4 amino acids, and this time we have a mutation here at position 4. We have a U instead of an A. As you can see up there, it was an A. That introduces a stop codon. UAG is a stop codon. That will tell the ribosome, hey, we're done here. Let's pack up and go home. Okay. But it results in, as you can see, 1 amino acid in this sequence instead of 4. And you look at that and immediately you say, nonsense. That is complete nonsense. There's no way that is right. So this is a nonsense mutation. So really that is the extent of what you need to know as far as this course for mutations. It's just a brief introduction to how this can affect how these problems in DNA and then also resulting in problems in RNA can result in problems in the protein sequence. So I hope this has helped you understand or better understand the content from our transcription, translation, and our mutation in this particular video. Let me know if you have any questions.